Well, I'm going to talk about mostly severe patients, and we talked about earlier that they haven't been studied very much. When we first started looking at research in this area, we decided to do severe patients because they have clearly something wrong with them. And looking at people with the most severe conditions will show us probably more details of what's gone wrong. The other thing that the principle was the fact that if you're going to study them, you shouldn't pick one thing at a time to study. You really should do the whole shebang. And that means collecting a very large amount of things and measuring everything you can imagine to be measured. So these severe patients that we analyzed have probably had more data collected on them than any other patient in the history of medicine. And we're still looking and we're still analyzing the data from them. Just to show you a severe patient, for those who haven't seen them, most people have not seen a severe patient because they never leave the house. They're bedbound, and this individual was part of the study. And I also want to use this the opportunity to thank all the patients that have contributed samples for all the different studies throughout the world to try to solve this disease. It's frequently very inconvenient to them, and sometimes even makes them worse. And so my gratitude to them. I just want to show you a few things. This is the severe patients, and in this study, and you can see that they were very inactive. In fact, this is from a Fitbit that was put on their wrist. Probably many of these supposed steps aren't really steps. It's a little jingling of their hand. And so, but we had to have some record that, in fact, they don't move around very much. So to do this kind of a study, we call it a big data approach, which is looking at a lot of things. And I'm not going to go through the details. I just want to show you a few of the things that we put on our list. We've added things to this list since that time, and are continuing to explore this possibilities. Now, for the first thing we focused on, in addition to just collecting the data, is so many patients have told me that they have frequent viral infections, and they get a relapse because they have a viral infection. And so we know how to analyze that. So I would like to find out whether that's true or not, or what is the virus that is in fact infecting them. And so we started out doing a possibility that there is a virus out there that has never been seen before. And that's always a concern because then everybody's going to miss it. And that was true for the HIV in the very early days. So there are ways to do that with current technology of sequencing. We have worked with Eric DeWitt, who's up in San Francisco, who gets blood samples from all over the world when someone has died what looks like a viral infection, but they can't figure out what the virus is. So we certainly don't want that blood in our lab. So he processes the blood to nucleic acid, which is then pretty safe, and sends it to us, and we analyze it. And the way you do that is that in the blood, there's an awful lot of human DNA, and so you first have to eliminate that because that's all you're going to see. And you look for particles. And the reason you have to do particles is you want to get rid of the human DNA, and the way you do that is treat it with an enzyme that destroys and digests DNA. And the particle protects it. Now that particle could be a virus particle, a bacteria particle, a parasite particle, and so forth. And so this has a limitation. It has to be a particle, and it has to be in the blood. But in most cases, when we have done this with a new virus, it does work. And so that's the first pass here, is to look for something new. And we didn't find anything. And the way you do that is you extract the DNA from the particle, and then you do massive sequencing. And you take every trace that you get, and you compare it against everything that's ever been sequenced in the entire world. And that's a massive computational problem. So we basically wound up totally occupying the Stanford cluster computer. Eventually we got kicked off because we were consuming almost all of its time and had to buy another cluster computer to do this. So that cluster computer runs 24/7 for these kinds of analysis. And we didn't find anything. Now, there's a nice built-in control because everybody in the world is infected with a virus called TT. We have no idea what it does. You get it shortly after birth from your mother, and everybody has it, and it's in your blood. And so if you don't find that, you did something wrong. But we decided we should actually do a more sensitive search, not for particles, but for a virus present in someplace else in the body that doesn't release particles into the blood. However, it's been pretty clear now that you have an infection anywhere in your body, the nucleic acid from the infecting organism will wind up in the blood. And in fact, as Ron Tompkins says, the blood is the sewer of the body. Everything gets dumped into it eventually. And so that's how you get rid of things. You put them in the blood. So in that situation, we have to make a very specific assay for that. And it's called polymerase chain reaction. It goes in and amplifies up the sequences, and then you sequence it. Now, the problem is that can get very expensive, so we have a really wonderful guy in the lab that can figure out how to do things much cheaper. In fact, I've spent the last 40 years making instruments that are smaller and also trying to do everything cheaper. And so every time we do an experiment, we worry about how to make it cheaper. And so in this case, he figured out how to do them all simultaneously in one tube. And it cost us $10. And so it's much, much cheaper. And basically looking at that, we looked at all the DNA viruses, and a couple of patients had a small amount of HHV6 and EBV, but so did the controls. In fact, the controls had more. So it is possible that they can get recurring viral infections, and it could probably cause them a problem, but it's not something that seems to be anything greater than what is found in healthy people. Well, the other possibility is a parasite. Now, it might not be particles that are found in the blood, so we looked at all the sequences for all the parasites. We made probes for them. These are parasites, trypanosomes, and so forth, with the idea that they will, if they are there, they will get in the blood. And it's certainly possible. And one reason for doing the parasites was looking at sleeping sickness. And if you look at the symptoms of sleeping sickness, I always thought it was because people slept all the time. No, it's not. It's called sleeping sickness because the patients with sleeping sickness are active at night and sleep during the day. And they're fatigued. They have absolutely all the symptoms of chronic fatigue syndrome and ME. So that suggested maybe there is a parasite that's throughout Europe and the US that people haven't, it's a trypanosome that people haven't found before. So we made probes for that, and that's still in the works, but we have not found anything. It's in the works because we've identified a few other parasites we didn't know about before that other infectious disease people said, so we have to add those and redo the experiments. We also want to do all the RNA viruses. That's hard because RNA is not very stable. So we decided to do the others first, and now we're working on the RNA viruses. I think we can do that. We also have to do all the funguses, and that's hard because there's an awful lot of funguses. We have to figure out which ones to do. And the bacterial pathogens are really hard because they're everywhere. Your skin is covered with them. If you take a blood sample, you invariably will wind up with some bacteria in it. So it's hard to sort out whether it's a contaminant or it's actually an infection. You also have leaky gut, and it's possible that DNA from the organism can get into your bloodstream. So we do find bacteria, but it's not clear it's a real, but it could just be a contamination. So this is typical of research. You try all these things and you come up empty. However, what we need in this case is sometimes negative results. And this is a case of negative results. Doesn't mean it's over with. You may find things you've done wrong and go revisit it. But at this stage, I'm not sure we should be looking for some new infectious organism unless somebody has some new ways to do it. I'm just going to show you a little bit of data from the severely ill, just to show you that they're very different. This is some sleep measurements, and between the healthy people and the patients, and they're very, very different. And obviously, anybody who's talked to a severe patient, they know they have real sleep issues. This is looking at the cytokines. At Stanford, we do a 63-plex losing Luminex assay. This was all set up by Mark Davis, and Mark has been a major collaborator with us for years now. And I just put up here in a circular pattern, healthy people, moderately ill, ME/CFS, and severe, and the severe patients, in most cases, have higher levels. If we just look at a few examples of that are very, very different, and I want to just point out a couple of things. One is that the cytokines might not be just high, sometimes they are low. And this was one that affects the brain, and it's very low, and it's not good to have it be low. And here's one, leptin, which has been known before, but it's high in the patients compared to healthy controls. So these are just indicators. You put this in a database for anybody to look at, and if they can make any connections to these. We haven't really followed heavily on the cytokines. I don't think that, from the data we have, that they can make a very good biomarker. You can also look at metabolites. We initially looked at 592 metabolites. These are all done by Metabolon. One of our severe patients was, I think, the first patient that Metabolon has ever done. And in fact, they had to do a lot of work on it because they hadn't done it before. They did it for free. They said they spent $16,000 on that analysis for free. And I just want to point out a couple of things that highlighted. We're now up to 850 metabolites doing this analysis. But here's a metabolite that's very low in the patients. And it's indole propionate. And it's a neuroprotectant. So that's critical that it's low. It's also a very potent antioxidant, and it's almost totally missing in the patients. Now you look at that and say, well, you first immediately say, oh, well, there's a genetic problem. No, the gene that makes this is not in the genome. It's in the microbiome. So this is coming from a bacteria in the gut. And as far as we can tell, that bacteria is missing in the gut. And it seems to be missing in every one of the severe patients. So there's a possibility here, as Reason pointing out, that this is something that you can chemically synthesize. I understand there are some companies that are now manufacturing this, and it has to go through clinical trials and so forth to make sure it's not toxic. The compound won't be toxic, but it could be contaminated. And this might turn out to be something that would help patients if you just simply put it in the food. It's already present in almost everybody, and it's coming from the gut, so putting it back in the gut should be okay. But it's something for a doctor to actually recommend and so forth. The hydroxyproline, which is very high, suggests that, and that's coming from digestion of collagen, suggests that there's a lot of degradation going on in the patients. Now, we did a whole genome sequencing of all the patients. It was a very high quality sequence. It was done for free by Illumina. And I'm just pointing out some of the key genes that turn out to be very different than the healthy controls. We're going to follow up on the KER genes. That's come up several times in different types of analysis, that it must be somehow critically involved. And it's like the HLA locus, which is your individual identity, but it is generally seen as controlling the sensitivity to viral infections. I'm not going to go into any detail of these genes, just showing you find genes, there are genes that are very, alterations in those genes are probably very significant. Now, this has been published, but I have a lot of questions, so I thought I would briefly mention it. Several years ago, on an NIH grant, we developed a device called the Nanoneedle, and the idea behind this was for doing diagnostic assays. And what we discovered is that and I won't go through the history of this, but we were working with cancer and other antibiotic resistance where we were using it. But then we decided to try to use it for a diagnostic tool for diagnosing ME/CFS. All this work, in fact, I'm talking about, it was all supported by the Open Medicine Foundation. It's a very small device. I have one here. So this is the chip that's used. We do a lot of measurements with it. These are the electrodes that are here. These are very small. They're 50 nanometers. That's about 1/1000th the size of a human hair. So you can't see it. It's on this silicon chip, but you can't see it. If you look under a microscope, you still can't see it because it's beyond the resolution of light. And there's two of these coming in, and then these make electrical connections. So we can measure the electric current going through this. And we measure electrical impedance. Now, that's an electrical term, and the reason we do that is it's very sensitive to changes. That's all. There's nothing magical about it. If the chemical composition of the cells change, you'll see it. If the size change, you'll see it. If the distribution of some organelles inside, it will change the impedance. That's all the reason we do the impedance. There's nothing magical about impedance other than it's very sensitive and easy to measure. And now we measure, we have 2,500 electrodes per centimeter, so there's lots of measurements going on. And we also make measurements 200 times a second. And we run this for several hours. So we have millions of data points. And for a diagnostic tool, that's actually very useful because so much diagnostic tools makes one measurement. And if there's a problem, you can get the wrong answer. And so with this kind of a measurement, you not only get a number, but you get the approach to that number. And that can often say, hey, this doesn't look right. There's something going on wrong. So it prevents you from making mistakes. And I'll just show you, this is the patients in the red. You get it, back up a second. We tried to see what would happen when we just put the blood in the sample, and nothing. They look just like healthy controls. So then we said, well, what if we stress the cells, just like the patients get stressed, although it's not really the same thing. Can these cells handle stress? And we want to put a demand on them to produce energy, because we had suggestions from the metabolomics that you weren't producing as much ATP, maybe, or NAD. These are critical energy components. So the real simple trick to do that is just simply put some table salt in. That's sodium chloride. Sodium is not a good ion for the cells. It will go into the cell, and the cell has to pump it out, or it'll be in trouble. And that pumping requires energy. So we put a little bit of sodium chloride in, and then they have to pump it out. And what happens is the sodium chloride is added over here, I think right there, and that's why you get a little glitch because of the sodium being added. And it takes a while before you get the response. And it's quite a bit of difference in time, and that might have to do with the health of the cell, I don't know. But what happens is you get a big change in impedance, and all that is that something happened to that cell that changed its structure. It could have actually died. That would definitely do it. But what's interesting about this is that there's just 20 patients and 20 controls. All the patients do this and none of the controls. And that was what we were after. Initially, we're not after saying, this is ME/CFS, nothing else. That's a very hard thing to do. But what we wanted to do is say, are they not healthy? This individual is not healthy. And distinguish it from a healthy control. And this does extremely well at doing that. And also the reason to do that is that so many patients who are sick go to the doctor, they run standard tests, the tests come back totally normal, and they tell you, there's nothing wrong with you. And that's crap, right? And you know it, that's the problem. And you can't convince the doctor because, oh no, all the tests come back normal. Now the picture I showed you of that patient that's severe, who cannot eat, who cannot drink, who has to be tube fed and a port into the heart to give liquid, cannot talk, cannot look at anybody. If you run those same standard tests, he's perfectly healthy. There's nothing wrong with him. So the tests that the doctors do to determine that are bunk. They'll catch other diseases, but they will not catch this, and that's the reason for doing something different. Now, I wanted to move on to sort of what's next, because that's what everybody asked me. And we have added 10 more patients and 10 more controls, and they look the same. And we'll continue to do that. Hardest thing we have in doing this kind of diagnostic thing is to make sure that the patients have the disease. And that's a real problem, because there may be other things that sort of look like it. And maybe they'll meet the criteria, the diagnostic criteria, but they don't have the disease. Well, then you'll get a wrong result. And it'll look like your test doesn't work well. No, it works perfectly. That's exactly what you want it to do. So we require to meet all the diagnostic criteria for each patient, and they also have to be seen by a specialist that's seen hundreds or thousands of patients. And they have to confirm, yes, I'm sure this person has ME/CFS. So that makes everything very slow. And so all these patients have been confirmed. Now the problem is the electronics box that we use for this is about the size of a big cooler. It weighs almost 100 pounds. It costs $30,000. This is not a good instrument to stick in like a doctor's office. It'll never happen. However, it's very sophisticated and we don't need all the sophistication. So what's next is to build a new instrument and not have it weigh 100 pounds. We want it to be about this size. And it's projected to cost about $200, which is what we do. Figure out how to make it cheaper, and it will also not, the big instrument will do 2 samples at a time. The new instrument will do 100. Now that's critical because we want to try to do some drug testing with it, and we need a higher throughput instrument to do that. We also have to figure out how to, we can't just give this chip to a doctor. You've got to make the electrical connections. Now the electrical engineers do it by hand. Not going to be possible. They do it by hand under a microscope. Nobody's going to do that. And I don't think even the researchers are going to want to do that. It's really hard. And so we have to figure out a way to mount this something that's very useful. And I have another possibility I'm going to show you. But the idea is to get this throughput up on this thing so that we can start testing drugs. Now those drugs, if they are positive in this assay, it doesn't mean what's going to happen in the patient. Oops. There are other possibilities that we are considering as well that might be useful. And this is one that we're probably going to do. It's something that was made several years ago. It's actually a flexible circuit board. And in that circuit board, we can measure things. And what we can measure is lactate, glucose, sodium, and potassium. And we can probably figure out how to do other things. But I'm interested in particularly is the lactate, because lactate seems to be high in the patients. And this might be a way to measure to prevent patients from overdoing it. We can stick an alarm on this, or in fact, we can give you a little electrical pulse to tell you stop. Now, it's a sweat detector, so it's non-invasive. That's the reason for doing it. It just simply measures all these molecules in the sweat. And of course, what patients tell me, I don't sweat, I can't get that active. No problem. We figured out how to electrically induce the sweat. Now, you can't feel it, but we can actually turn on your sweat glands. And so we can collect the sweat when we need to make the measurements. So this is a possible thing we're considering measuring, and probably couple this with other things like heart rate and everything else. And we may deal with some manufacturer of these devices to put this all together. That's a possibility we're looking into. Another thing is a technology that we developed some years ago that we put it on the shelf simply because we had no grant money to pursue it further. And this is a little electrical device, and it's very cheap. So what you're seeing here is electrically operated cell sorter. Cell sorters cost about $100,000. And how it's made is a little sheet of plastic right here. And instead of doing the standard fabrications, we simply make a two-dimensional inkjet printer that you can buy at any store. And we take out the ink cartridges and put in silver nanoparticle ink, which you can buy. And when you print that, it makes an electrical contact. So you then can design it so that it, in fact, makes all the electrical circuits that you need, and you don't have to have an electronics box. Now, the nice thing about this, two nice things about this cell sorter costs 1 cent. So when you use it, you can throw it away. Secondly, the design of this can be made anywhere and emailed to anybody. So you could, anybody in any place in the world could make it. You can get, all you need is a printer and the ink. So that's something that we'll pursue. We have to see if we can make the same kind of test. We don't know that yet. So we have to figure out which of these things to pursue. And I just want to tell you what's next. But I also wanted to show you, we've found 2 compounds that affect the nanoneedle assay, and it make it look like normal. And these are just things that have been suggested to us by patients or things that we have decided to look for. And we're just beginning because we don't have high throughput. But one of the interesting ones is a compound called Copaxone. It's an MS drug. And the design of this MS drug is to make the site the antibody recognizes. And so the idea is to make a decoy. And it has these 3 amino acids in it, randomly polymerized with random length. It's pretty simple. We're now making some in the lab. And it has to be put in by injection, so that's the inconvenience of it at the moment. But when we put it into this blood sample from a patient, it looks healthy. And there's another compound called SS31. It's going to be approved soon, and it's for mitochondrial damage. And that also makes it go flat. And I wanted to show you that I put up the structure. It's also 4 amino acids, but notice that, in fact, it has tyrosine, which Copaxone had, and it has lysine, which Copaxone has. And it's 4 amino acids. So maybe there's something about that we have no idea why it works. So we're going to start making compounds with these amino acids and see if we can find a pattern. The other thing is to make another device that we could also diagnose the patients. And one of those is with the red cells. And we've detected that red cells are not as deformable in the patients. That could affect blood flow. There's a number of other diseases that have deformability problems, namely things like sepsis, and that relates back to what Ron Tompkins was talking about with the trauma and so forth, and all the expression and changes that he sees. And the device then, here's one here. This is one of the devices we can use. Again, it's kind of small, cheap. I think this is going to be it's probably going to be 10 cents, a little expensive. But you flow the red cells through, and you photograph them, image and analyze them, and they don't go through as quickly. And that's because they're not as deformable. And so the blood flow may be less. And here's some of the devices. This is all done by computer simulation initially. I'm going to run through this. But this shows you deformability of a healthy control is high. It can be really deformed. and the ME/CFS blood samples are not. The variance, we measure 1,000 cells and do the image analysis on that, so that's why we have a pretty tight variance on this. So last year I talked about a model that might explain the disease, and it was called the metabolic trap. Now, we thought we would figure this out fairly quickly, but we've had a lot of, and what you do in this is you don't try to find data that supports it. That's cherry picking, that's a no-no. You'll get the wrong answer if you do that. What you have to do is to try to disprove it, which is what we've been trying to do, and we haven't been able to, but we don't trust the data. So this involves predictions that are made by the model, and then we measure them in the mass spectrometer. The problem has been the cells that are potentially trapped, these are cells that make the kynurenine pathway, is very small. There's only about 1%. So I'm just going to move on. Oh, this is an instrument you can actually buy that looks at the blood flow. I'm going to go through this. These are just showing you some toxic stuff. Whoops, there we go. So we haven't been able to, we get the measurements that say it's consistent with the hypothesis, but the error in the measurements is too high in our opinion, and we don't want to trust it. So we have to figure out a way to get the signals higher, and that's what we've been spending our time on. It's only about 1% of the immune cells have that pathway. So we have to purify those cells out, and then also the media that is around commercially is screwing it up. And so, and then we have to make new media that's not commercially standard media. And then we have to get onto a very high-end mass spectrometer, and that's very hard to get onto. Everybody wants to use it. So in the meantime, how this all started, this hypothesis started, was from collecting all this data. And we were going through the data, and we were looking at the genetics of things that we thought, and this is with Robert Fair was doing this, and that could be key to the disease. And one of them was looking at the kynurenine pathway, because the kynurenine pathway, which makes kynurenine, which makes NAD eventually, which is involved in energy production, and it also makes serotonin, which is in the brain and in the gut. And so, well, that's a candidate. Let's look at that. And what they discovered in our 20 severe patients is, well, let me go through and just show you one other thing. I'm going to back up. Here's the pathway. Here's tryptophan, and tryptophan is converted to, through a couple of enzymes, to serotonin in the brain. But tryptophan is also converted to kynurenine, and kynurenine is a major regulator of the immune system. So this makes it interesting from that point of view. But the interesting thing about this is there are two enzymes that do exactly the same thing. One's called IDO1, and the other is called IDO2. And what the geneticists and biochemists have said is that, well, this is an older enzyme, it has lots of mutations, so it's irrelevant. You shouldn't even be working on it. It's IDO1 that's an important one. So when we did the sequencing, we found that every patient had a mutation in this other enzyme IDO2. Now that was, and we found it because we were doing it by hand. If you do the automated analysis, it doesn't show up, simply because it's excluded by the software because it can't possibly be important because it has too many mutations. And so they don't tell you that you have mutations. And so, and that's because 75% of the population has a mutation in IDO2. So if IDO2 is really important as a susceptibility, it means 75% of the population is susceptible to ME/CFS. That ought to scare people if they really know what this disease is. So we decided, because we're trying to get the mass spectrometer and all that, well, we'll just sequence more. Now, that's expensive. The whole genome sequencing of the quality we did here is about $1,000 a patient. So we decided to do it differently, and we got our guy that can do all this magical amplification stuff. And so he worked out a way to sequence this gene for $10. And so we then sequenced 46 more of the patients that we were sure had the disease, which is what was useful for having this analysis, of making sure they have the disease. Otherwise, we could get confused. All 46 have the mutation. So that's 66 sure patients have a mutation in IDO2. Now, the way to think about that is that having a coin. Now, coin is 50-50, not 75, so it's not perfect, but it gets close. To flip a coin 66 times and have it come up heads every time. Now, you can do that in your head, say that's not likely. Right. And so the same is true here. It's a little more likely, but it's not very likely that that could happen by accident. And we're going to do more to make it even better. But the probability that that's the case is, I think, 0.0003 or something like that this would happen by chance. So what that says either two things, that IDO2 has to be involved in this disease, or the data are wrong. You always got to consider that all the time. So this is the pathway. Now, how in the world so you have these two genes. This is the one that's defective. It has a lot of mutations that are found in the population. So why in the world does that have to be important? The biochemist and the geneticist said that can't possibly be important. And that's what Robert Fair figured out. And the reason he figured it out is he went back to a paper that was published in 1967, which no medical student will look at because it's too old. And that is that, here's the paper, and that says that the other enzyme, IDO1, is substrate inhibited. What that means is the substrate's tryptophan, which is the amino acid, an essential amino acid, you can't make it. If you get the tryptophan too high, the enzyme shuts off. And it makes kynurenine. The only way to get it back on is to reduce the tryptophan. The only way to reduce the tryptophan is to have the enzyme active. So you are trapped. And if you do modeling of that, it's very unlikely you'll ever get out of it. Now, in the body, there's a backup. And that backup is IDO2. IDO2 is not substrate inhibited. It doesn't work very well. It works at very high concentration of tryptophan, but it's what gets you out of the trap. And this is just what happens to the activity of the flux through the pathway. Here's what happens with IDO1, and then it gets substrate inhibited, and now you can't make kynurenine. And then if you have IDO2, they work together, and it looks like a normal enzyme reaction. And you're out of the trap. So that's where we are. The genetic data says IDO2 must be involved, and it's really hard to figure out how in the world can you have an enzyme where 75% of the people don't have it be so essential. I know this is fairly complicated, but this disease is complicated. It's likely the solution is complicated too. Now, the wonderful thing about it is that this pathway is on in dendritic cells. Dendritic cells control the immune system. There's papers now that show that in order for the dendritic cells to control the immune system, they have to have the kynurenine pathway on. This says if you're trapped, it's not on. It will not be able to control the immune system. It will not be able to suppress autoimmunity. There's also, this pathway is on in the serotonin-producing cells in the brain. Where are those cells? In the brainstem. With the pictures you just saw. There are not very many of them either, but they make serotonin and melatonin. And so, and there are also the pathways in the gut. And serotonin is needed in the gut. So it would explain why you have brain effects, immune effects, and gut effects. Now, the problem is, this is the bad news. How do you get out of it? Now, I immediately said, well, fine, you just don't give them tryptophan. Get it out of the diet. It doesn't work. And the reason it doesn't work is that the only way to get rid of the tryptophan is for the enzyme to be active. You can't, the enzyme, the transporter that transports it out of the cell only works at very high tryptophan. And you can't get the tryptophan low enough by that transporter. So we have to come up with some very novel solutions. But we're working on it. So it means a drug. It means a drug that will block the inhibition. I'm going to stop there.